Welcome to MedTechISO. I am your host Arvind Rathore, and in today's video, we are focusing on one of the most important aspects of ISO 109931 compliance, choosing the right biological test. With so many testing options like cytotoxicity, sensitization, irritation, and more, how do you know which one are necessary for your medical device? In this video, we will provide an overview of the key biological tests, explain their purpose, and discuss the criteria for selecting the right test for your device. Let us get started. ISO 109931 provides a framework of biological tests to evaluate how medical devices interact with the human body. Each test is designed to assess a specific type of biological risk. Let us briefly go over the most common tests. The number one is cytotoxicity testing. Cytotoxicity is one of the most basic tests and is almost always required. It evaluates whether the material in your device are toxic to cells. In this test, cells are exposed to extracts of the device and their viability is measured. If the cells die or functions are impaired, the material may not be the biocompatible. The number two is sensitization testing. This test checks whether the device can cause an allergic reaction, particularly in individuals with sensitive immune systems. Sensitization is especially critical for devices that contact the skin or mucosal surfaces, as these are the most common points of allergic reactions. The number third is irritation testing. Irritation testing assesses whether the device can cause local irritation when it comes into direct contact with tissues, such as skin, eyes, or internal surfaces. This is particularly important for devices like wound dressings or the surgical gloves. Number fourth is systemic toxicity testing. This evaluates whether the device or its leachables can cause harm to internal organs or systems after being absorbed into the bloodstream. Systemic toxicity is crucial for implantable devices or any device with prolonged body contact. Number fifth is hemocompatibility testing. For devices that contact blood, like catheters or the stents, hemocompatibility tests ensure the material does not cause harmful effects such as clotting, hemolysis, destruction of red blood cells, or platelet activation. Number six is xenotoxicity, carcinogenicity, and reproductive toxicity testing. These tests focus on long term risks. Xenotoxicity checks if the material can damage DNA, while carcinogenicity assesses the potential to cause cancer. Reproductive toxicity evaluates whether the device can impact fertility or development. The number seventh is implantation testing. Implantation tests are conducted for devices that are placed inside the body, such as pacemakers or orthopedic implants. These tests evaluate how the surrounding tissues respond to the device over time. These are just a few of key tests outlined in ISO 109931. The full list can seem overwhelming, but here is the good news. Not every test is required for every device. The selection of tests depend on the nature of your device and its intended use. Now that we have covered the types of tests, let us talk about how to determine which ones are relevant to your device. ISO 109931 emphasizes a risk-based approach to testing, meaning you only conduct the tests that are necessary to evaluate the specific risks associated with your device. Here are the main criteria to consider. Number one is nature of body contact. The first and most critical factor is how the device interacts with the body. Is it in contact with the skin, blood, or internal tissues? For example, a device that only touches intact skin may require irritation and sensitization tests, but might not need systemic toxicity or the hemocompatibility test. On the other hand, an implantable device would require a more comprehensive evaluation. The number two is duration of contact. ISO 109931 classifies devices based on their contact duration. Limited exposure, which has less than 24 hours, prolonged exposure, which has 24 hours or 30 days of exposure, permanent exposure, which have more than 30 days of exposure. Longer exposure durations typically demand more rigorous testing. For instance, a temporary catheter may not require a xenotoxicity testing, while a permanent implant would. Number third is material composition. The materials used in your device heavily influence the testing requirements. If your device uses well documented biocompatible materials with a history of safe use, you may justify waiving certain tests. However, novel materials or complex composites will likely require a full battery of tests. Number fourth is type of use and environment. Consider the device's functional environment. Devices used in sterile internal environments like vascular stents require more stringent testing compared to those used externally. Similarly, devices subjected to mechanical stress like orthopedic implants may need additional tests to evaluate how material degradation impacts biocompatibility. Number fifth is regulatory expectations. Different regulatory bodies have varying requirements for biological testing. For example, the FDA may require additional tests for certain device type compared to the EU MDR. Always align your testing strategy with the regulatory framework for your target market. Number six is risk assessment results. A thorough risk assessment can help you determine which tests are necessary. If your risk analysis identifies potential concerns such as leachables or degradation products, those specific risks should guide your testing plan. Now, here are some practical tips for choosing tests. When selecting biological tests, it is important to work closely with a multidisciplinary team, including material scientists, toxicologists, and regulatory experts. Start by conducting a material characterization study to understand the chemical and physical properties of your device. Use this data to identify potential risk and tailor your testing strategy accordingly. Always document your rationale for selecting or excluding specific tests. Regulators will want you to see clear justification for your decisions. For example, if you waive xenotoxicity testing because the material has a long history of safe use, make sure this is well supported by scientific evidence. Finally, don't forget to consult accredited testing labs with expertise in ISO 10931. These labs can guide you through the process, ensuring your tests are conducted according to good laboratory practices and meet regulatory standards. To summarize, choosing the right biological tests for ISO 10931 compliance involves understanding your device's body contact exposure duration, material composition, and functional environment. ISO 10931's risk based approach ensures that you conduct only the necessary tests while avoiding unnecessary costs or delays. By carefully evaluating the biological risk of your device and tailoring your testing strategy, you can ensure compliance while prioritizing patient safety. If you found this video helpful, please like, share, and subscribe to MedTechISO for more detailed insights into ISO standards and compliance strategies. Have questions about selecting biological tests for your device? Drop them in the comments below. I'd be happy to help. Thank you for watching, and I will see you in the next video.